here, and I'm really happy to be introducing Reid today. And Reid's father, Ben, is also in the audience today, so welcome. So Reid is a biochemistry major and has 2 minors in neuroscience and interfaith leadership. Beyond all that, Reid has spent several semesters with the Spiritual Life crew, which has been a big piece of her Wilson experience. Let's see, Reid's project, well, first let me say, upon graduation in May, Reid hopes to eventually go to grad school in microbiology or public health. And more importantly, according to Reid, she wants to have a yard big enough for chickens and a garden to feed her neighbors, okay? All those goals, okay? So this is a new project in collaboration with Melanie Wilder at Fiber Arts, and it's utilizing Wilson-grown indigo. And so the project is trying to get at how bacteria allow us to dye cloth with indigo. So Reid used a lot of new techniques, had to do a lot of troubleshooting, method optimizing to do this. Great work, excellent, excellent to work with. I really appreciated it. So thank you, Reid. Take it away.
Thanks, Kim. I'm going to try to project. Welcome. On the slide here, we have a picture of a young indigo plant growing down at the Warren Wilson Garden during the summer of 2020 when they were harvesting indigo. Melanie's back there tending to the little shoots. In recognition of this land, I'd like to read this land acknowledgement that allowed us to grow these indigo plants. So, we reside on Salagua-Tiwi, home of the eastern band of the Cherokee Nation, who were forcibly removed from this land in order to make way for settlers of European descent. This work could not have been done without the land, which grew the indigo plants and which does not belong to us. Furthermore, we are informed as craftspeople by indigenous craftspeople of this region and all around the world, whose methods and craft call us into deeper stewardship to all of creation. May all of our work honor and recognize and encourage the return of this land to its rightful stewards. So indigo, or indigotin as the molecule is technically called, is a blue dye. It is, at least originally, plant-derived. Pictured here is Persicaria tinctoria, one of the plants that we grew down at the garden and that indigo can be derived from. As a dye, it is, or the molecule itself is insoluble in water. Pictured on the left is indigo powder settling out of water during a extraction technique, and therefore it's called a vat dye, meaning that it needs to be solubilized in some form or some way in order to dye cloth. So many things are blue. I'm wearing a lot of blue today just by accident. And one of our most common blue dyed things that we think of is jeans. But jeans these days are not usually dyed with plant-derived, plant-done indigo. But this on the right is shibori, which is a traditional Japanese use of indigo dye and resist dyeing. If you want to know more about it, you should talk to Melanie, not me, about resist dyeing. But there's interest in returning to a more traditional use of indigo. So the Japanese method can be illustrated like this. So on the left, we have a Japanese indigo plant, which is different than the one we grew on campus, but has this precursor to indigo that is endogenous to the plant cells. Through oxidative fermentation, you arrive at tsukumo, which is the name of the composted leaves in the Japanese indigo process. The indigo in the leaves is insoluble, and through a fermentation process, which is that bacterial metabolism of the plant material, you can arrive at an indigo vat that contains leuco-indigo, which is the reduced form of the molecule. And through oxidation, you can return back to indigo once it's been fixed on the fabric. And there it's fixed to fabric and insoluble in water again. So in a redox reaction, you have something being oxidized and something being reduced. Confusingly, the thing being oxidized is called the reducer, and the thing that is being reduced is called the oxidizer, but those are details. And so in the indigo situation, you begin with indigo and you end with leuco-indigo, which has this very greenish hue to it. And in the case of indigo fermentation dye vats, there's a bacterial addition of this redox enzyme that helps along the process. So like I said, most commonly these days, indigo dyeing is not done via bacterial reduction. Most often the indigo is reduced using synthetic chemicals, of which sodium dithionite is one example. And so it can replace that bacterial reduction, but of course comes along with its own set of consequences, most of which involve sodium dithionite being still pretty active even in the wastewater of those sorts of systems. And we don't want a ton of it in the environment. So if we're wanting to return to natural indigo dyeing processes, there's interest in how those processes can be optimized and simplified so that they maybe don't require the centuries of learning to care for them that is currently the case. In pursuit of answers to this question, I know et al., a research group in Japan, as well as some others, have elucidated connections between oxidation and reduction potential, which is the potential of a system to reduce things, dye readiness, so when the vat is ready to dye things and make them blue, and the bacterial genera that are present in the vat at a certain time. Seen here is a graph showing the pH of the vat and the ORPs, that oxidation reduction potential, over time. So for our research, what is more common for artisan work is what, for my purposes, I called an artisan fermentation vat, which is begun with indigo powder and not the composted leaves, and importantly is much smaller than the traditional indigo vats. So these artisan vats can be done in 5-gallon buckets, not huge sheds size kind of vats. So here you can see indigo powder, and then some indigo powder samples from the plants, the various plants that we grew down in the garden. So my question was, what are the bacteria in these kinds of vats, these artisan fermentation vats, and how do the vat conditions correlate with the readiness of that vat to dye things blue? So pH, ORP, those sorts of conditions in the vat, and what are the bacteria that are present in those vats? So in June of 2021, for 2 weeks, I ran 4 fermentation vats that looked like this. They were bubble wrapped, and underneath this contraption in the biochem lab, to be insulated well, to maintain a warm temperature. I dyed small cloth swatches to see when the vats were ready. That's how artisans determine when the vat is ready, is just by testing small bits of cloth. And I acquired those probe data, so oxidation reduction potential, pH, and temperature. And I also collected fluid samples, so a little bit of this liquid, in order to see and see what bacteria were there. So one set of cloth swatches looked like this. And so over time, there is more reduction of the indigo, and so more dyeing of the little swatches. The swatches looked basically like day 2 before they were put in the vat. And the stars here indicate which days that we carried through with the bacterial isolation. Due to material and time constraints, we didn't do that for all 6 of these days, but for 2, 6, and 10. The results of the probe data look like this, which is similar to the graph from before from Ino et al. The ORP drops over time and can be seen correlating with the dye capability on day 6 from this previous slide. And the pH drops a little due to acid production. If you were to maintain the vat for weeks or months, you would maintain the pH at a high alkalinity. So for each of those sample days, I removed a little bit of fluid and diluted it and plated it. And after 3 days at 30 Celsius, the plates looked something like this with individual colonies. And the colony indicates that there was one bacterial cell that landed on the plate at that time and multiplied and grew up into a visible colony. So how might you go from colony on a plate to identification? You go, you amplify DNA through polymerase chain reaction, which is also a whole class in genobiology. And then that DNA is sequenced, and you enter your DNA sequence into a database of ribosomal RNA sequences, which are unique to each bacterial species. And then you generate a list of things that it is likely to be marked by percentages of matching. This is what my isolation and sequencing scheme looked like. So for days 2, 6, and 10, I isolated 24 bacterial colonies, sequenced 19, 22, and 19 of them, and in the end, came away with 7 unique isolate identities for days 2, 6, 2, and 6, and 4 unique isolate identities for day 10. Pictured here are my BLAST-IN is the name of the database used to compare sequences. And on the left are the species that matched with my various bacterial samples, isolates. The percent matching says how many of those base pairs matched with the cited sequence. And relative abundance out of 60 is for 4 of the 60 sequences that I entered into the database. They matched most closely with S. agaricans, and so on. This one is highlighted because it has the lowest percent matching, and it is well under the threshold for identifying bacterial species. So it is and there are a lot of them. So it's likely that this is something new, and it's probably not that. But there would be, I only did partial rRNA sequences, and so there would need to be more work to determine what this isolate might be. These pie charts demonstrate those same results, but by day. So day 2 and 6, there were 7 unique isolates, and day 10, there were 4. This suggests that there may have been a relative abundance of strains decreasing over time, but more work would have to be done to solidify that claim, as I was not doing any quantitative isolation of bacterial sequences. So our next step was trying to test if any of those could possibly be reducing the indigo in the vats. The indigo was reduced in the vats. That's what our swatches showed us. And so seeing if any of our isolates had done that. So we used indigo carmine plates to begin that process. Indigo carmine plates are blue when you make them. And it's a similar enough molecule to indigo that it can be a sort of precursor for seeing if you should continue with study. So halos on these plates would indicate degradation of the indigo carmine. So you can see in the circle there are 2 different isolates that have little yellowish halos around them. So each of these streaks are one isolate. Once we had 11 isolate matches, we inoculated indigo containing cultures with these 11 species. So this is especially difficult to see in this light, but these little tubes have culture broth in them and a little bit of solid indigo, and were inoculated with the isolates. And the green hue that is in some of them would indicate indigo reduction in that small tube. They're like little miniature indigo vats. So here are my indigo reduction results versus previous literature. So these are the isolates that I isolated that indicated some form of indigo degradation or reduction. And here are these 3 have been previously isolated from indigo vats. And E. polygamy is a known indigo reducer, cited by Ino et al. As indigo reducing. And then these 4, interestingly, indicated to me some sort of indigo reducing capabilities, but have not been previously isolated from indigo vats. So characterization of these bacterial isolates is space for further study. What can we learn from them? What was allowing them to thrive in the indigo vats? Are they reducing indigo? Which of them are creating the environment that allows others to reduce indigo? Just learning more about them. It's also interesting to think about how the traditional methods might be different than artisan methods. Do they contain similar bacterial genera, or are they very different? Do the outcomes differ significantly, and why might that be? Another interesting future direction that we were not able to do in this study is an ecological succession of bacteria in the vats. So which bacteria are suited for the vat at a certain time and not later in the process, which of them thrive when the vat is stable, and which of them were there right when it began and sort of petered out. This flowchart is what I did. It's colony-dependent methods. So of all of the bacteria in the vat, was only able to pick from the ones that grew on a plate. And of the ones that grew on the plate, I only picked 6 of them. So I'm missing a huge number of the things that were in the vat. So this study was a preliminary investigation, hopefully the first of many, and I pass on the torch to those who come to study these kinds of vats further. I'd like to thank Kim for being my advisor, and Jeff and Elisa for being part of my committee. Melanie Wilder, thank you so much for your expertise and time. I'd like to thank the Sutherland family for funding this study, allowing me to get necessary materials. My partner, friends, and family for being here for the whole year. And really grateful to the indigo plants and this land for growing them. Other questions? Yeah.
Do you think it's possible, since indigo grows on many other continents and culturally indigo plants all over the world, that there would be specific bacteria in the places that have indigo plants that would be unique to different places?
Yeah, I think that's highly likely by my estimation. All of my yeah, I think that's a really interesting direction, and definitely all of my research and sort of literature study was on mostly these groups in Japan, studying Japanese indigo plants. And yeah, there are indigo precursors in plants that grow mostly in Europe, Eastern Europe. And so yeah, I think that's highly likely that there's really are region-specific bacteria, as well as some consistency across that. The bats are a pretty particular kind of environment, and so I think it's also likely that there's kinds of bacteria or genera that are well suited to the alkalinity and the temperature and that kind of thing. Yeah.
Thank you so much, Reid. This is fascinating and so cool, and I hope just the beginning. You left us with so many cool questions too, and really cool answers as well. A question I have, the ecologist in me, right?
Yeah.
is really excited. I was so glad you mentioned that at the end. And I guess I'm wondering about artisanal practices, and that I noticed in your pie charts, some things pop up at day 6, right? That weren't there, and you alluded to that. Whether even the artisanal practices of what you test with, I'm thinking of, I think contamination has the wrong, whatever that word is, I'm really trying to start with. But anyway, gives the wrong impression that it's negative, but just reasons you might introduce new types of bacteria along the way, right? Connotation, that was the word I was looking for. There it is. Anyway, right? So where was my brain just now? Anyway, question about the artisanal processes and introductions of bacteria along the way.
Yeah, there's that's definitely something that that this literature talks about, is that these vats, the traditional vats, last for like a year or more, and are maintained, and many of them are like people get in them and like store them around with their bodies, like people dip all kinds of things in there, and they are pretty amazingly steady across that time. They require a little bit of mixing so that nothing like hangs out in one corner, but other than that, they are pretty consistent. So I guess one thing that would be interesting for smaller vats, it seems like those would maybe be overtaken easier by something coming from the outside that doesn't fit in well. But that's definitely another like really interesting question, is what is the succession of bacteria, but also what's the community at the end when they're stable that allows them to, maintain and not get overrun by contamination. Jeff like one time said something about like you could put kombucha under your bed and just leave it, and it actually doesn't get that gross. It like does its own thing, and I've thought about that a lot since then. This idea that there's something in there, it's maintaining itself,
Right.
and that's sort of the succession, but also a kind of another thing too.
Yeah, stabilization of the community. Cool.
Yeah.
Thank you. Anything else?
Yeah. Yeah, we did.
I guess a couple of questions. One following up on that, like pH 10, 11, I mean, that's kind of wild in itself if they're so alkaline loving. That probably has at least a little bit to do with it. Are there other, has anyone looked like other archaea or other sorts of microbes that might be good? And then one other unrelated question is, so the indigo gets reduced into that. Is it just oxygen that oxidizes it back, or okay?
Yeah, so you when that vat is active and looks kind of green and has this shine on it, you dip your fabric in, and when you the other option for this picture was a video of me dipping something in and watching it, because you can watch it turn blue.
Oh, really?
Over time.
Oh, wow.
So you and even with like the little swatches, you put them in and they look kind of gray-green, and over the course of 5 minutes or so, they oxidize back and look blue. So as far as I know, it's just oxygen. And then, yeah, I don't know if other people have done, if people have studied other microbes. That's definitely one of the connecting pieces between all the ones that have been found is even the ones that I found that haven't been previously isolated, were like isolated from like marine mud. So other places that are high alkalinity and are obviously well suited to being in that kind of environment. But yeah, I'm not sure about other microbes. Yeah, Greg.
Yeah, I was wondering, what was the material of that, like the vessel itself, what was made out of, in your experience?
Well, the vats I did were just like rubber, not rubber, plastic buckets, yeah.
So like in traditional areas where they have these giant vats, they generally made out of like pottery or wood?
I think probably like cement-like things or pottery, that kind of thing. The like, that picture that was at the very beginning of the Zukomu, they're often done in these like almost shed-like things to be composted in. But yeah, I think
So are
ceramic.
they used batch to batch, is the same vessel used batch after batch?
Yeah, so that's another, yeah, these vats that are traditional vats are just like run over and over and over again. And so you can, I guess similar to your kombucha SCOBY, keep adding to it. So even with these artisan vats, you feed them, you have to give them wheat germ or other sorts of things like that and maintain their alkalinity or give them more of the composted leaves, and they just keep going.
The reason I ask is just because, like, you have a certain like microbial profile in the lining of the vat itself. Because, like in Japan, like the best soy sauce is maybe seen in these
Yeah.
wooden vats because of the microbes that exist, you know, for decades and decades in the wood itself.
Yeah.
So I wonder if that might influence like these places that make the good indigo. They
Yeah.
have these ancient vats that they
Yeah,
have cured over time. Blah, blah, blah.
yeah, that is interesting. Who's next? Mark, maybe?
Well, I was curious about the pH, like Lane was asking too, but this high pH, and maybe I missed it, but I mean, does the pH go up because of the bacteria, or is there something added to make the pH high so that bacteria live in there? And kind of
Yeah.
related to that graph from Anina you showed, was interesting because the pH started high,
Yeah.
it went way down, and then it went way back up again. I was like, is that just a poor pH measurement, or did it really do that?
Yeah.
So,
Yeah, so you start the vats very alkaline, so they're started with,
the lye,
they can do lime and blanking on what I started with. But yeah, so this one, you can see the ORP is this bottom one.
Yeah.
And it drops dramatically right at the beginning. And then their pH, they're maintaining it. So these arrows indicate addition of Lixivium, I think, to keep the pH really high, which I wasn't doing, because I just did those 2 weeks and it starts to drop a little.
But see, the pH actually started really high too. There's a dot up at the.
Yeah,
That's what
so-
I didn't get. It went really high and it dropped dramatically, and then all of a sudden it gradually creeped up.
Yeah, so I'm not sure about why it would have dropped so dramatically at
Yeah.
first. But yeah, it was there. The traditional vats also are started with soda ash or something else to make them highly alkaline at the beginning. Yeah.
I'm curious about the genetic differences, since I know you did some PCR between the original Japanese indigo and the American indigo that you studied. Was that a part, looking at the differences in those two plants, it sounds like it was part of your study. Were you able to have any genetic look at the comparison between the two?
I didn't look at the plants at all. I just, I started my vats with indigo that was extracted here. But yeah, I didn't look at the differences between the plants at all. Yeah.
So were the microbes doing this, were they from the soil, or were they from the plants, or from the air? I was a little confused on it.
Yeah, at least from the literature, the idea is that they're coming, in the traditional vats, coming from the Sukomu, so coming from the composted leaves. It's not clear here where they would have come from. maybe from the indigo powder itself, that it was started with.
So you
or-
use distilled water or something, put the dye in and then
Yes.
chemicals to make it alkaline?
Yes. Any other questions? Okay, so let's then create a form for everybody.